morphologic, clinical aspects, and extrolite production. You know, Aspergillus flavus group is an extremely important genus concerning pathogenicity, mycotoxin production, and useful extrolites. Aspergillus oryza and Aspergillus soja are thought to be domesticated variant of Aspergillus flavus and Aspergillus parasiticus respectively. Aspergillus section flavi now comprises 11 closely related species producing different secondary metabolites. As you know, we have aflatoxigenic species and non-aflatoxigenic species. When we know that Aspergillus oryza and Aspergillus soja produce useful extrolites from ancient. And Aspergillus flavus and parasiticus, you know, they are mycotoxigenic. So these days we use molecular methods, and we have some Aspergillus that use, for example, you see Aspergillus sodotamari, Aspergillus nomius, Aspergillus sodonomius, Aspergillus menesclerotigenes, Aspergillus novaparasiticus, and like this. You know, every microorganism goes to these by molecular methods. Aflatoxin toxicity in human can produce hepatic and gastrointestinal injury. It can be immunosuppressive, can teratogenic, it can be teratogenic, can be oncogenic, and if used more than 2 to 6 milligrams per day per month, it can be severe liver injury. And medical mycologists know that Aspergillus possesses many enzymes, many metalloproteins, many pectinase and amylase that it can harm the mammals. It can invade the mammals, the insects, and produce some injury in plants, in cotton balls tissue. And in human, we have invasive and non-invasive aspergillosis by Aspergillus groups. And it can produce allergic bronchopulmonary aspergillosis in asthmatic patients, in cystic fibrosis patients. The pathogen can attack any part of the body, sinuses, can skin and heart, lung, kidneys, and anywhere, like produce keratitis on the eye and can produce onychomycosis on the nail. In certain geographical locations like Saudi Arabia, Sudan, and Iran, my country, with semi-arid and arid dry weather condition, the disease caused by Aspergillus flavus is more common than that caused by Aspergillus fumigatus. You see in the text, the more you are familiar with Aspergillus fumigatus, which is, the virulence of it is most than Aspergillus flavus. But in this area, like my country, Aspergillus is more. And it was accounted for more than 80% of the total Aspergillus infection in keratitis. As I said, several species has been described in the past which are assigned to Aspergillus section flavi, mainly based on traditional method. When you use traditional method, like morphological parameters, like colony texture, and sizes and texture of conidia, conidiophores, and structures, you can detect them by morphological method. Recent data indicate that several of the species assigned to section flavi cannot be distinguished based on morphological features alone, and they use molecular methods. This is Aspergillus flavus. It has been isolated from my patient. This is one isolate, but on different culture media. It is at the left is at CYA, Chapex yeast agar, at 37 degrees centigrade, that at 25 degrees centigrade, at Chapex yeast sucrose agar, at Chapex dux agar. You see the different colony texture. And the back of the colonies of these are all one strain I isolated from patient. You can see the grooves, the radiate grooves on these, they are different. And this is this is three isolates that I then, after I did molecular methods, I identified that there are Aspergillus minesclerotigenes. Aspergillus minesclerotigenes is very new. By molecular method, they identified. I can say that I found when I worked on morphological, when I worked them, I can guess that they are a little different from the other Aspergillus flavus I worked. This is Aspergillus oryza, which they used for useful extrolites. I had isolated this isolate from the sinus, a patient from a sinus, and it was identified by Parvin Dehghan, the one who is mycologist, knows Parvin Dehghan in Australia, food science. This was, I confirmed that is Aspergillus oryza, that's from sinus. The structure on the microscope, you know Aspergillus had a conidiophore, a vesicle, biseriate, one seriate, phyolite, and conidia. And Aspergillus sexin flavi had equinolate or rough conidiophore or a steep. This Aspergillus reference strain, it was my reference strain, and that is the three Aspergillus that I said. They are a little different from others. I want to say one thing, that environmental factors is very important in the texture of colony, in the metabolites that it produces. The different metabolites can act on different physiological effect. You see the different physiological metabolites that they produce. For example, carbon, glucose, and sucrose, simple sugars like glucose, sucrose, will increase the aflatoxin biosynthesis. And nitrate will decrease the aflatoxin biosynthesis, and the ammonium will increase it. The pH, optimum pH for aflatoxin synthesis is 3.4 to 5.5, and the temperature is 29 to 30 degrees for optimum condition for producing aflatoxin synthesis. And the water activity, you know, below 0.85, the growth of the fungus, and it is S4 germination rates will decrease. So, and aflatoxin production is significantly decreased at temperature below the 25 degrees centigrade, but it's completely, they say, it's completely inhibited at 37 degrees centigrade. I want to say that a standard culture media, like malt extract agar, at 25 degrees centigrade for morphologic identification we should use 25 degrees centigrade and malt extract agar. If you use any other culture media, the texture will change a little. Aspergillus, different Chapex agar will differentiate between the Aspergillus species. Aspergillus flavus and parasiticus agar. This is an screening culture media for detecting Aspergillus flavus and parasiticus from the soil or from the nuts, from the seeds. You can use Aspergillus flavus parasiticus. And if you want to screen if one Aspergillus can produce aflatoxin, you can use coconut cream agar for screening. I used all of these with molecular methods. I used morphological methods, phenotypic methods, and genotyping methods. That is the one that is there. That is Aspergillus flavus parasiticus agar. If it produces the fungus produce orange color, it is Aspergillus flavus. They say Aspergillus flavus. Pitt says it is Aspergillus flavus or Aspergillus parasiticus. But in the isolates I have isolated from patient, we had pale colored. Don't produce aspergillic acid, which produce this this orange color. And this is the malt extract agar, and this is coconut cream agar that I say for screening. You can see the blue fluorescence or white fluorescence around the colony behind a very simple device that I will show you. If you use different culture media, I said the colony texture will change. That is Chrome agar candida. Chrome agar candida, the Aspergillus flavus is with the blue ring, and Aspergillus oryza with a like this colony, you can see. Cutty says that Aspergillus flavus isolates can be characterized by two distinct strains, S strains and L strains. S strains, they are produce small sclerotia, and L strain, they are that produce large sclerotia. I say it depends on the culture media you used. It is sclerotia, small sclerotia, and that is large sclerotia. All of these pictures are from my isolates that I worked on them. That's sclerotia. It's sandy like, I don't know who knows, who has seen. It is, you can see with naked eye sclerotia on the plates. This is a smaller sclerotia. This is one sclerotia that I measured the diameter of them. I have used V8, and I had a big sclerotia, large sclerotia. And on CYA, I had small sclerotia. I showed this picture. That was number 6 Aspergillus parasiticus, and that is one of my isolates. And this is the simple device that you can screening if this Aspergillus can produce aflatoxin or not. It is a lamp, a wavelength, 365 micrometers of ultraviolet UV. And this is HPLC. You know, this device is very complicated, and it can certainly, we can measure a little amount of aflatoxin by this. You know, all moldy grains, moldy pistachio nuts, they are, when you use butter, butter or milk, they goes to milk, to body fluid, anywhere. When you use moldy and nuts that they have Aspergillus flavus and produce aflatoxin, they go to body fluids. They hydroxylated in the liver. It can go to milk, it can go to urine, and it can go to peripheral bloods. It can go to meat of poultry, meat of calf, and we eat that. So, detection of mycotoxins themselves must be distinguished from the detection of mycotoxin-producing fungi. Method for detection of toxin or identification and isolate with a recurring emphasis on improving the speed and lowering the cost of the assays. In general, the aflatoxin gene cluster in Aspergillus parasiticus and Aspergillus flavus consists of 25 genes spanning approximately 70 kilobase pairs. And that's interesting, I say, Aspergillus flavus, Aspergillus parasiticus, Aspergillus oryzae, and Aspergillus soja all have some of these genes, and a key gene, AFLR. I found that all of these isolates have the AFLR gene, a key gene for producing aflatoxin. The FDA has set limit of 20 parts per billion total aflatoxin for interstate commerce of food and feed, and half of a part per billion for aflatoxin M1 for sale of milk. Aspergillus soja and oryza are widely used in koji preparation in the Orient. In the Orient, Aspergillus nomeus is morphologically similar to Aspergillus flavus, but produces series B and G aflatoxins. That's Aspergillus oryza or soja. That soy sauce is a popular worldwide condiment that originated in China somewhere between the 3rd and 5th centuries. It is made from the paste of boiled soybeans mixed with some type of roasted grain, normally with barley or rice, brine and salted, mixed with water, and Aspergillus soja or oryza molds, and it left to ferment for several months, after which it is pressed and strained before being bottled. And we have soya sauce. Aspergillus section flavi include now more than 25 species, and this number is likely to increase significantly in the near future because of increasing application of the phylogenetic species concept based on DNA sequencing data rather than on visible morphological characters. Recently, a 6-step molecular strategy used real-time PCR, rapid PCR, ESMA1, digestion of the nuclear DNA has been worked out to distinguish 9 of these species. The genomic size of Aspergillus flavus is bigger than the Aspergillus fumigatus, and it is believed the latter, means Aspergillus fumigatus, has lost some parts of its genome during the lifetime. In conclusion, despite intensive investigation, the taxonomy of Aspergillus is still highly complex. Recent data indicate that several of the species of this genus cannot be distinguished based on morphological or molecular method alone. However, no single method can be used to classify Aspergillus section flavi. Molecular identification should be compatible with morphological feature and combination of identification method should be used for satisfactory results. Thank you for your attention.